In this video, we will discuss about the mechanism of the reduction in the dopamine in cachexia conditions in the cancers. So, in order to find out the mechanism, they developed some in vitro system where they introduced this, use this VTA and have this NAC and directly checked this firing rate of dopamines in the pre-cachexia and the cachexia conditions. And what they are able to find out is that in this case they are able to see that the dopamine formations when they use this and in order to check this cachexias in the real time, they use this AAV retro YFP virus. And what they are able to find out is that there is a continuous decrease in the firing rate of dopamines. And when they check the in the panel B, this shows the overall RMP in the millivolt, and here we can see that the difference is statistically significant between the pre-cachexia and the cachexia conditions about the dopamine levels. However, the other parameters such as this amplitude and this IEI, amplitude remains non-significant, but this IEI signals is highly upregulated in the case of cachexia conditions compared to the non-cachexia conditions. And thereafter they did some literature studies and able to find out that before these cachexia conditions affect the dopamines, there is a GABA pathways that can also get disrupted, and that can also get disrupted in the cachexia conditions. So the hypothesis that it's the GABA receptors that is creating a suppressions in the dopamines. So this is the immunofluorescence image, and here what they check is they check this GABA receptors that triggers this SNPR that triggers these GABA receptors. And what they are able to find out, wherever these SNPRs are present, they are able to see more suppression in the dopamines. And this could indicates that during the cachexia conditions, these SNPRs, which are usually present in the PBNs, gets triggered, and which has an inhibitory effect on the dopamines. So in the panel F, they have shown the mechanisms of how these things are interrelated. That is, ARP will act within this PBI at 3 different regions, that is VTA, SNPR, CEA, and BNST. The neuron formations are usually formed using this VTA. However, during the cachexia conditions, these SNPRs gets activated, and it has more inhibitory effect towards these neuron formations by the VTA. These things they have checked, and these things they have checked this inhibitory reactions formations, that is the activations of these SNPRs during the cachexia conditions is significantly higher, both in the case of amplitude and also in the case of frequency. Thereafter they decided to check whether it's this reductions in the dopamine is because of SNPR or not. So they used the 2 different strategies. First one is by using this crimson TDT, thereby they suppress this, prevent this SNPR activations, or we can see that the GABA receptors get inactivated using this crimson TDT. And thereafter they check the excitations to inhibition ratios, and they can see that excitation to inhibition ratios goes down in the cachexia conditions significantly compared to the pre-cachexia conditions. And then from the other approach that they used is use this ex vivo slice GAD to CRE, and this they use it in the form of lentivirus, in the form of virus. I'm not sure which kind of virus they used, but they prepare a virus of this gene of interest, and then check and inhibit these GABA receptors, and then check the percentage depressions. And in terms of the amplitude, they do not see a significant difference, but in terms of the frequency, they can see that there is a significant upregulations in the case of cachexia conditions compared to the pre-cachexia conditions. So indicating that it's the GABA receptors that get disrupted during the cachexia conditions that will has a inhibitory effect on the neuron formations by the VTA. So this is the mechanisms that they are able to find out from this results that are presented in this figure. If you need to understand more about their mechanisms of reductions in the dopamines in Cancer-Cachexia conditions, you can go through the reference that has been provided in the description, and how they come out to these GABA receptors is also shown in the supplemental figures. So it's worth visiting this paper to understand this concept in depth. Thank you for your time.